till the finish line. I have my private practice at a place called Palakkad in Kerala. So today I will be giving you a very brief introduction about blood physiology with a small insight into the red blood corpuscles. Right? So when we talk about blood, blood is the liquid connective tissue of our body. The fact that it is liquid ensures that it reaches every nook and corner of the body. And that is so important because every nutrient that we ingest, the oxygen that we breathe in, the carbon dioxide that we release out, all these things have to be taken to and from every cell in every corner of the body. So it's very important that there should be a medium which collects all these things from one site and takes it to another site and replenishes the stores of the cells at a regular period of time. This helps to maintain the viability of a cell throughout its life cycle. So when we talk about blood, it is often considered to be the fluid of life. That is because of the simple fact that it is involved in the transport of nutrients, vital nutrients, and oxygen to different parts of the body. And this is very important for the viability of a cell. It's also known as the fluid of growth. The reason for this being it is important in transmission of hormones which bring about changes at sites which are very distant from the site of secretion. That is, for example, your thyroid hormone. It's produced at a particular site, but its action translates to different cells at distant locations in the body. That's an example. It's also called the fluid of health. The fluid of health for the simple reason that it carries immunoglobulins and white blood corpuscles, which help in maintaining the defense system of the body against various pathogens. So it's known as the fluid of life, growth, and health. Coming to the general properties of blood, it's scarlet red in color in case of arterial blood, and it is usually purplish red in color, which is known as the venous blood. In general, usually arterial blood contains oxygenated blood. An exception to this is the pulmonary artery, and venous blood carries deoxygenated blood. An exception to this is the pulmonary vein. The volume, the total volume of blood in the human body is about 5 liters, and it's an alkaline fluid with a viscosity which is 5 times that of water. Now we'll just have a brief overview about the components of blood. Now, though blood is a liquid connective tissue, it has two parts. One is the part which forms the liquid component, which is the plasma, and then you have a group of cells which perform specific functions at different points in time. These cells are called the formed elements. The cells comprising the blood are collectively known as the formed elements. Formed elements comprise of red blood cells, white blood cells, and platelets. In the white blood cells, you subdivide them further into granulocytes and agranulocytes. This differentiation is based on the presence or absence of certain granules with specific functions in the cytoplasm. Then you have the platelets. The platelets are basically involved in the clotting mechanism of the blood, also known as hemostasis. The plasma, on the other hand, forms the liquid component of the blood, comprising predominantly of water, which is about 92%, and about 8% solids. Among the plasma proteins, you have albumin, globulin, and fibrinogen. Okay, albumin is a simple protein, which is about 4.7 grams percentage. Globulin is about 2.3 grams percentage. And then you have fibrinogen, which plays a very important role in the clotting mechanism or hemostasis, which forms about 0.3%. So all in all, you will observe here that the albumin to globulin ratio is about 2:1. This is a very important fact. It has been asked multiple times. So you need to remember this. Okay. So what you should remember here is when we take these short sessions, it's very important that you pay attention to each and every slide because we are trying to condense the information which is present in the textbooks, which is much more elaborate, into a much more concise form. So listening to 15 to 20 minutes of an online session is equivalent to reading a textbook for about 1 to 2 hours. So it's very imperative that you keep your ears wide open and all your sense organs totally alert. Okay? So remember here, the albumin to globulin ratio is 2:1. Now we come to the topic of the day, the RBCs, or the red blood corpuscles. Now, the red blood corpuscles are a very, very fascinating aspect of physiology. When you talk of physiology, the beauty of physiology lies in the fact that you are able to translate everything that you do on a daily basis, something as simple as breathing or talking. You're able to translate that right down to the cellular level. So when you talk of the general function of blood, it is so multifaceted. So many things are happening at the same time. You are able to trace all of that back to a cellular level. And here, one of the most abundant formed elements in the blood is the RBC. Okay, the RBC is unique in that it is more of a vessel than a cell. It is unique because it is a exception. It's an exception to the general eukaryotic cell. When you talk of a eukaryotic cell, you're talking about a cell which is having abundant organelles, a well-defined nuclear aperture, and it is able to multiply and undergo cell division at a regular rate. But that is not the case for an RBC. An RBC is unique because it has depravity of organelles. The regular cell organelles that you see in a cell are not present in an RBC. We'll come to that subsequently. Okay, it has a diameter of about 6.9 to 7.4 microns, and it has a biconcave shape. Note the diagram here. See this, it's central depression with a peripheral extension. It has a biconcave shape, as a result of which it has a thickness of about 2.2 microns at the periphery and 1 micron centrally. So it's compressed towards the center and is dilated towards the periphery. It has a surface area of about 140 square microns and a volume of about 85 to 90 cubic microns. Now, the important thing here is the biconcave shape of a normal RBC. Now, this is very important because this shape is ideal to allow the cell to permeate into the thinnest of capillaries. Had it been round or of any other shape, the problem is that it is going to get stuck between the capillaries, the very thin capillaries, and this in turn can cause obstruction of those capillaries and eventual lysis of the RBCs. So the very purpose of the RBC being a vessel to transport certain substances to different parts of the body is defeated. The second thing is this wide shape gives it abundant surface area. It reduces and increases this, it increases the surface area, which allows it to take up maximum amount of nutrients and distribute it to the distant sites of the body. So this is very, very important in pertinence to the shape of the RBC. Now we come to the formation of RBCs. The RBCs are also known as erythrocytes, and hence the formation of RBCs is called erythropoiesis. Poiesis means formation, erythro means red. Okay. Now, erythropoiesis occurs in 3 distinct stages in the prenatal life. Okay. The first stage is called the mesoblastic stage. The mesoblastic stage occurs in the mesenchyme of the yolk sac. The mesoblastic stage occurs in the mesenchyme of the yolk sac during the first 3 months of intrauterine life. The second stage is called the hepatic stage, wherein RBC synthesis or the erythropoiesis shifts to the liver. This occurs up to the 6th to 7th month of it, from the 3rd month onwards, up to the 6th or 7th month of intrauterine life. Then you have the myeloid stage. The myeloid stage starts from the last 3 months of the intrauterine life and continues throughout life. This myeloid stage is where the RBC synthesis occurs within the bone marrow. Okay, so these are the 3 subdivisions based on the stages at the location at which RBC synthesis occurs. So you have your mesoblastic stage, you have your hepatic stage, and then you have your myeloid stage, 3 stages. After this, you have postnatal, that is after birth. After birth, RBC synthesis is confined to the bone marrow. Okay, during the first 5 years of life, all the bones will produce RBCs, all the bones in the body. From the 6th year of life right up to the 20th year of life, you have erythropoiesis occurring in the long bones and in the flat bones. After the 20th year of life, the RBC synthesis is restrained or restricted only to the flat or membranous bones. This is because the long bone marrow, the bone marrow within the long bones starts being replaced by fatty tissue after the 20th year of life, and hence they lose their erythropoietic function and it is restrained only to the flat or membranous bones. So these are the sites for our erythropoiesis. Very, very important. You should remember this. So let's revise that once again. Prenatally, you have your mesoblastic stage, hepatic stage, myeloid stage. Mesoblastic, restricted to the mesoderm of the yolk sac, first 3 months. From the 3rd month onwards, it's the hepatic stage where the liver takes over. And then in the last 3 months, you have the myeloid stage where the bone marrow takes over. Again, after birth, you have 3 stages. The first 5 years, it's in the all the bones of the body. From the 5th to the 20th year, it is in the long bones and the flat bones. And after the 20th year, it is restricted only to the flat bones because the long bone marrow is replaced by fatty tissue. Is this clear? This is the outline for the site of RBCs. Coming to the process involved in RBCs synthesis, which is erythropoiesis, it occurs under the influence of a hormone which is called erythropoietin, which is basically a glycoprotein in nature. It's an erythropoietin. Erythropoietin is produced in the capillaries in the kidney, okay, in the renal capillaries. So erythropoietin brings about the stages of erythropoiesis. Now look at this diagram here. I want you to focus on this part of the diagram. Okay, this will elucidate the changes that are occurring in a precursor cell so as to allow it to differentiate from a pluripotent stem cell to a mature erythrocyte. Okay, this is very, very important. You should understand that the synthesis of the formed elements of the cells, formed element cells of the blood occurs through pluripotent stem cells. Pluripotent means plural, means multiple, potency. So they have multiple potencies. That is, a particular cell is having the ability to give rise to different types of cells based on the stimulus and the requirement of the host. Got this concept? So here you have a colony forming unit here, CFU, or the colony forming unit for erythrocyte, which gives rise to the precursors for the erythrocytes. So here you have the proerythroblast, the early normoblast, the intermediate normoblast, late normoblast, and the reticulocyte, which in turn gives rise to the erythrocyte. Now the important thing here is, look at the diagram here. It starts turning. You have a large cell which is blue in color. See the blue cytoplasm here? Yes, it's hematoxophilic. And as you go down, it starts becoming eosinophilic and the organellar component starts reducing. So from a mature, premature normoblast right up to the mature erythrocyte, the only change that you're going to see is a reduction in size, reduction in organelles, and an increase in pigmentation. Very, very easy. That will help you to summarize the entire process of erythropoiesis. So you have your proerythroblast. Always a blast cell means a precursor cell. So look at this, it's a proerythroblast, large cell with a blue cytoplasm and a large nucleus. Then you have the early normoblast. Again, slowly the colors of the cytoplasm starts changing. And then you have the intermediate normoblast. Intermediate normoblast. Intermediate normoblast is very important because this is the stage of erythropoiesis where hemoglobin starts appearing for the first time. Hemoglobin starts appearing for the first time in the intermediate normoblast. This is the reason why the intermediate normoblast is also called a polychromophilic normoblast. Polychromophilic. Why? Poly means multiple colors. Chromo means Poly means multiple and chroma means colors. So polychromophilic normoblast. That the reason for that being In general, the cytoplasm is hematoxophilic or blue in color. Okay, but due to the appearance of hemoglobin in the intermediate normoblast stage, the cytoplasm starts becoming slightly pinkish in color. So you have two colors within the cytoplasm, hence the name polychromophilic normoblast, right? Then you have something called the late normoblast. Late normoblast is characterized by the absence of organelles, and you'll have a small remnant of the nucleus, which is called the pycnotic nucleus or an ink spot nucleus. Pycnotic means the nucleus resembles an ink spot. So you'll have a distinctly hematoxophilic point in the cell, which represents a remnant of the nucleus. Now here, slowly the color of the cytoplasm is pretty much eosinophilic because hemoglobin takes over most of the cytoplasmic component. So this is known as an orthochromic normoblast. The late normoblast is also known as the orthochromic normoblast. Then you have the reticulocyte. Reticulocyte is the immediate precursor to the mature RBC. Right? Reticulocyte usually forms about 1% of the RBC component, which is about 4.5 to 5.5 million cells per cubic micron, which is the normal RBC cell population. 1% of that is formed by the reticulocytes. Reticulocyte, the reason for that is because these nuclear and cellular remnants form thread-like extensions throughout the cytoplasm. That's why reticule. Reticule means finger-like or strand-like extensions. Cyte means cell. So a cell exhibiting finger-like or strand-like extensions, hence the name reticulocyte. And then you have finally your mature RBC as we know it, as we saw previously, the biconcave shaped cell that is devoid of all organelles. This is very, very important. This is the exception to all eukaryotic cells. This RBC or mature erythrocyte is a cell that is devoid of nuclear and cytoplasmic organelles. The reason for that being its main function is that of a vessel. It has to carry something from one place to another and back. That's it. Okay, so it's very important that it should have a very, very frugal nutritional requirement. That is, it should not have much of a nutritional requirement. It should be able to sustain itself on very, very minimal nutrients. The reason for that being, had it been having all the nuclear organelles and on nuclei and all other cytoplasmic organelles well intact, then it will start utilizing all the substances that it is carrying for its own work, isn't it? For its own metabolism, isn't it? For example, if you ask a hungry cat to carry a bowl of milk from one room to another room, do you think that bowl is going to reach the other room with the milk in it? No, isn't it? It's going to drink that milk because it needs to sustain itself first. That's the same thing applies to a cell. If you give a cell nutrition, the first thought that comes into its mind is to prolong its survival or to facilitate its sustenance. For that, it will use all the nutrients which is present in it. So to avoid this, the RBC has been kept devoid of all organelles. Because of that, it has a very, very limited nutritional requirement, and it has glycolysis to provide for its ATP requirements, and hence it is able to transport almost all the nutrients in its nascent form right to the site where it's required and back. Okay. Coming to an important component in the cytoplasm of the RBC, which is called hemoglobin. Now, hemoglobin is a conjugated globular protein which has a molecular weight of about 64,000 Daltons, right? And you should remember the values of hemoglobin. It's very, very important. The quantity of hemoglobin decreases from birth till adulthood. So it starts from as high as 20 to 25 grams per deciliter, right up to 14 to 16 grams per deciliter in adults, right? So it's usually in a range of 14 to 16 for adult males and 12 to 14 grams per deciliter in adult females. So the derivatives of hemoglobin comprise of the different components with which hemoglobin can attach itself in the RBC. So if it combines with oxygen, it's oxyhemoglobin. With iron, it is ferrohemoglobin. If it combines with carbon dioxide, it's called carb hemoglobin or carbamino hemoglobin. Very, very important. This is very important because you need to make a distinction from carboxy hemoglobin. Okay, carboxy hemoglobin is where carbon monoxide combines with hemoglobin. Carbamino hemoglobin or carb hemoglobin is where carbon dioxide combines with hemoglobin. Okay, carbon dioxide combines with hemoglobin in a normal condition during the transport of respiratory gases. However, carboxy hemoglobin is usually seen in pathological conditions, especially in carbon monoxide poisoning. It's very important to remember that the affinity that hemoglobin has to carbon monoxide is about 200 times more than that it has for oxygen. This is why the moment a person is exposed to carbon monoxide in very high concentrations, he can perish very easily because the stable bond that hemoglobin shares with carbon monoxide ensures that this bond remains irrevocable, and hence there is no oxygen supply to the host, and hence he asphyxiates and dies. Right? If it combines with sulfur, it's called sulfhemoglobin. With nitrous oxide, it's called nitrous hemoglobin. And a condition where the hemoglobin is reduced, in a reduced form, it's called methemoglobin. Okay, so these are the derivatives for hemoglobin. Now you have the structure of hemoglobin. Hemoglobin comprises of two basic structures, as you might have seen in the name, heme, globin. Heme is the pigment, which is the iron containing pigment, and globin is a protein. Globin is the protein. Okay. Now there are double bonds which are present in heme, and this is what makes heme into a red color. That is what gives heme the red color. Okay. Every molecule in hemoglobin combines with and is capable of combining with oxygen reversibly. This is very, very important. I cannot overemphasize this enough. You should remember that the bond which hemoglobin has with oxygen is totally reversible. Why is this of physiological significance? Because if oxygen combines with hemoglobin irreversibly, how is it going to release it into the distant cells? Isn't it? It's going to keep it to itself. It's not going to share, isn't it? So as a result of that, you will have a hypoxic state. So to prevent that, it has been designed in such a way that the affinity which oxygen molecules have to hemoglobin is very, very limited. This is why the combination of oxygen with hemoglobin is called oxygenation. Okay, it will remain in an oxygenated state, not oxidation. Oxidation is much more stable. Oxygenation is a little more unstable. So it is able to release this oxygen into the nearby cells. So this is how the synthesis of hemoglobin occurs. Okay, it usually occurs within the mitochondria, characterized by the combination of succinyl-CoA with glycine, giving rise to pyrrole. And four pyrrole rings together give rise to a structure called protoporphyrin. Protoporphyrin, among them, protoporphyrin 9 is the one which combines with iron to give rise to heme, and heme combines with globin to give rise to hemoglobin. So here you have to remember it is protoporphyrin 9 which combines with iron to give rise to heme, which combines with globin to give rise to hemoglobin. Okay. Now here you should also remember that iron forms an important part in hemoglobin. Iron in normal hemoglobin usually exists in a ferrous state. Okay, it exists in a ferrous state, and this allows easy uptake of oxygen. Iron in the human body comes from ingested food, which is absorbed in the duodenal enterocytes, right? And it is either stored in a form called ferritin, or it is circulated in a form called transferrin. Trans means transporting, ferrin, iron, hence the name transferrin. And storage, ferris is the scientific name for iron, isn't it? So it keeps ferris in the blood. So ferritin, keeping the iron in the blood. So that's the storage form of iron. So ferritin and transferrin. Also, you should remember that 1 milligram of iron is excreted per day. Now, the destruction of RBCs. The lifespan of an RBC is about 120 days. After that, the cell starts losing its shape and normal contour, and the membrane pliability of the RBC is reduced. The flexibility associated with the plasma membrane of a cell is reduced. So because of that, it undergoes lysis. Now, this lysis can occur at two sites. One is extravascular, which is not within the blood vessels, and intravascular, which occurs within the blood vessels. Extravascular hemolysis usually occurs in the splenic capillaries. Okay, so how this occurs is there is an increased blood volume or a stagnation of blood which occurs in the spleen, as a result of which glucose levels reduce, glycolysis reduces. When glycolysis reduces, the nutrient supply for the RBCs is reduced, as a result of which oxidation of iron occurs, and maintenance of iron in a ferrous state becomes very, very difficult. When this happens, normally, in a normal RBC, iron is maintained in a ferrous state because this enables optimum oxygen uptake. When it becomes difficult to maintain iron in this ferrous state, what happens is the cell starts using all its ATP reserves, and hence the sodium potassium balance is lost, as a result of which there is an influx of water into the cell. When there is an influx of water into the cell becomes more rounded in appearance, as a result of which it gets entrapped within the splenic sieve, or the thin splenic capillaries, the thin capillaries within the spleen. When this happens, it gets ingested by the splenic macrophages, and this is the reason why the spleen is called the graveyard of the RBCs. Very, very important. Very simple, but very important. So here you should just remember that once the flow of the RBCs in the blood is suspended or obstructed, this leads to stagnation. When stagnation occurs, the RBCs are unable to facilitate their nutritional requirements and their metabolic requirements to maintain certain ions like iron in their ferrous state. Okay, so when this happens, it has to utilize its ATP reserves and hence undergoes a stressful environment. When this happens, the sodium potassium balance is lost and more water molecules come in from the extracellular fluid into the cell, causing it to swell up. And this in turn prevents it from squeezing through the thin splenic capillaries, hence entrapping them and subjecting them to phagocytosis by the splenic macrophages. This is the reason why the spleen is called the graveyard of the RBCs. Next, you have intravascular hemolysis. This occurs as a result of RBC destruction due to certain turbulence within the blood flow. This can be due to mechanical or traumatic factors. This can be something as simple as a turbulent blood flow, okay, or it could also be due to a blunt trauma to the surface. So as a result of this, RBC destructions can occur right at the site where the intravascular trauma has occurred. Now, there are two important proteins here called haptoglobin and hemoplexin, which help to salvage the iron which is released from the hemoglobin. So as you can see, nothing is going to waste. If hemoglobin gets broken down, globin goes back into the protein reserve, and in heme, the iron is separated out from it and utilized as a storage byproduct for subsequent RBCs and for subsequent synthesis of hemoglobin. Hope this is clear to you. Okay, so now let us see a few questions and then wrap up today's session. During the first 3 months of intrauterine life, yes, where does erythropoiesis occur? Okay, I need some answers from you now. Come on. Yes. Very good. It's in the mesoderm of the yolk sac. Mesoderm of the yolk sac, followed by the liver, followed by the bone marrow. Graveyard of the RBCs, yes. Graveyard of the RBCs is the splenic capillaries, very good. That's because of their thin diameter, they get entrapped there. Whenever you see a question like this, please let me get this straight right now. You're starting your preparations. Whenever you get a multiple choice question, if you have answered the option correctly, do not close the question at that. Okay, when you know the answer is splenic capillaries, you should revise the extravascular hemolysis flow chart that we saw previously. You should be able to trace back the reasons for the hemolysis. Okay, so how it is getting stagnated, how glycolysis stops, how it becomes difficult to maintain iron in its ferrous state. Yes, how it is causing the cell to utilize its ATP reserves, how the sodium potassium imbalance occurs, how water ingresses into the cell causing it to swell up. All these things should come back to you. Okay, if not anything else, you should also see the other three options, not in this case but in other questions. You should be able to relate to the other three options as well and study around the question. This will help you save a lot of time during your preparations. Okay. Which subtype of protoporphyrin is involved in the formation of hemoglobin? Yes. Protoporphyrin 9. Excellent. Protoporphyrin 9. Good. Okay. Variation in shape of the size of the RBC results in size. Size of the RBC is anisocytes. Isocyte means similar size. Anisocyte means variation in size. Variation in shape is called poikilocytes. Variation in size is called anisocytes. Variation in shape is called poikilocytes. Just look at these images. These are examples of poikilocytes. Look at this. Spherocytes. See these round cells? They're exactly round. This is called spherocytes. Then you have the sickle-shaped cells. Note this sickle-shaped morphological alteration here, caused by a mutation in the beta chain of hemoglobin. Yes? Substitution of valine for glutamate at the 6th position of the beta chain of hemoglobin gives rise to sickle-shaped cells. Then you have these teardrop-shaped cells. Look at the cell here. Teardrop-shaped cells. These are also called dacrocytes. Dacrocytes means teardrops. Teardrop-shaped cells. Hemoglobin first appears in which stage of erythropoiesis? Yes, come on. Yes? Where does hemoglobin first appear? You know it. Come on. Yes, good. Polychromophilic normoblast. Polychromophilic normoblast is the other name for your intermediate normoblast. Correct. Intermediate normoblast. Basophilic normoblast is the other name for early normoblast. Orthochromic normoblast is the other name for, yes? Orthochromic normoblast is the other name for late normoblast, isn't it? That's the stage where you see the pycnotic nucleus. Okay, good. Iron in hemoglobin is found in which form? Good. It's in the ferrous form. That enables easy uptake of oxygen. Ferrous form. Very good. The main protein involved in erythropoiesis is secreted from? Yes? Good. The peritubular capillaries of the kidney, peritubular renal capillaries. And what is that protein? What is that protein? Erythropoietin. Erythropoietin. Good. Erythropoietin. Nice. Megaloblasts are precursors for? Megaloblasts are precursors for? Normoblasts. B. Precursors. Precursors is a cell which precedes. So usually your precursor cell will be large when we talk of erythropoiesis. So a normoblast is something which is of comparable size to the normal RBC. A megaloblast is something which is really large. So usually the immature RBCs will tend to be, which are very early in their stage of development, will tend to be much larger than normal. So this is a macrocyte and a macrocyte. Look at the size of these cells compared to this. See, large cells. with thin central pallor. A microcyte, on the other hand, is very, very tiny. Look at the size in comparison to these cells. They're very small. Okay, so these are your microcytes and macrocytes. The normal shape of an RBC is, yes? Normal shape of an RBC, very good, very good. It's biconcave. This is what facilitates increased surface area for absorption, easy penetration through thin capillaries. Good. Pycnotic nucleus is a feature of very good. Orthochromic normoblast. Orthochromic normoblast, which is your late normoblast. Good. So this diagram will help you just recap your erythropoiesis very, very easily. It's a very, very simplistic diagram, kind of crude also, you may say, but it tells you everything you need to know. See the shape of the cytoplasm, see the staining of the cytoplasm, see the size of the cell, see the presence of nucleus, see how the nucleus is disappearing and how the changes in the staining pattern is occurring. Okay, so this is what will help you summarize it. Okay, thank you. So that concludes our brief webinar of the day. Hope it was useful for you and hope it gave you a brief insight into the world of RBCs and hematology. Thank you for your patient audience.
Thank you, sir. Thank you all for attending our webinar. And next webinar will be on cranial nerves by Dr. Pravish on July 27. So hope we can all meet at that time. Thank you, sir. Thank you.
Yes, thank you. Good night, everybody.